We will go on with the next speaker, and this is Professor Wolfgang Friess. He is the chair of the Pharmaceutical Technology and Biophysics, Biopharmaceutics, not biophysics, at the Ludwig-Maximilians-University in Munich in Germany. And he will talk about the use of lysophosphatidylcholine for the stabilization of pharmaceutical proteins, such as monoclonal antibodies, against absorption and aggregation.
Okay. Although I'm used from the lecture theater with the students that I could also do it without the microphone, I will use it. Well, good morning, everyone, from my side. What I would like to introduce you is now to shift gears, because we learned a lot about fundamentally the biologic use and physics of the lysolipids. And, well, the question, what can we use them for? And this was our thinking. Can we use the lyso PCs and the materials for stabilization of proteins? And I would like to give you a brief introduction. What is the background of that? Why we want to do that? And let's say, well, what is also kind of a difference than what we so far heard. So to set the stage, yesterday we heard, well, what is the future? And where's pharmaceutical industry supporting us, and why not? And I think we had fascinating results that we learned from the checkpoint inhibitors. So looking at the top-selling drugs at the time currently, we see that, let's take out the 2 vaccines, we have 5 out of these 10 are monoclonal antibodies. And this is, still a little bit lacking behind, because we are learning a lot from the formulation of these biologics. And they are, from our perspective, also already complex products compared to small molecules, and many of the aspects we can translate between, let's say, colloidal stability of something which is a protein, which may be a 10 nanometer size, which is already, let's say, nano, and what we learned from different or larger structures in this nano region. So the challenge is that we have these extremely successful products on the market. We have many of these molecules in the pipeline, but we still have, as the pharmaceutical industry would not call it problems, we have challenges. So the challenge is that these proteins are always considered with respect to stability, probably in the financial aspect ranging of a CCC rating of Argentina or Angola, because we have a colloidal instability, we have conformational instability, and we have chemical instability. So different aspects of that are that the chemical stability we have to look at, and not forget that these, say, the proteins, it's still chemistry behind. We have a lot of deamidation, oxidation, changes that can occur. Conformational stability may, in the past, have been, it's more or less, let's say, not of a problem for monoclonal antibodies. We have unfolding temperatures in the 60s degrees C region, should not be an issue. However, we may have different domains which are more susceptible or less susceptible. And we are also have a lot of interest in the colloidal stability, meaning the protein interactions, and in the end, potentially leading to protein aggregation, meaning whatever size of a structure which is larger than a monomer. It ranges from a dimer, goes into something which is in the micrometer range, and it could be something which is visible. And this is a problem. So one aspect of this aggregation phenomenon is that the proteins tend to aggregate at interfaces. The reason being that you see on the left bottom side, really when you have in lung trough your probe, and you can really dip your probe into this protein film. And this is not milk. This is an antibody solution that you can see here. And what happens is that protein adsorbs at the interface. So we have a highly concentrated stage at that point. And then the next thing is what we learned is that upon compression and decompression of that phase, then we even put them more together and stuck them together. And then we relax them. And then they say, okay, what are we going to do next? The molecules start thinking. So, and then they think, well, let's stick together. So we are an aggregate now. And this is causing problems because these aggregates then turn up in the drug product. The product comes into the patient, and the patient may, for example, it's not about let's say, inactivity more or less. It's only a tiny amount, but let's say the biggest concern is about antibodies forming against your drug and making it inactive. So what is the solution? It's adding a surfactant. So the surfactant can keep the proteins away from the interface. So what is illustrated here is that you have the different sizes of aggregates that can form, ranging from large visible particles to what you can see in the size exclusion chromatography to something in between. And you have to consider them all because protein may form dimer, it may form the large aggregates only. It's very specific, it's unique for the formulation as well. And when you shake it, let's maybe focus most on the shake, the white graphs, then you can see that, for example, shaking this formulation, for example, forms a lot of dimers in the size exclusion chromatography on the left bottom side. And when you add polysorbate, higher concentrations, you can resolve that issue. The other one that we found out, and then we even started to patent, is why don't you just simply fill up your container completely, consider a pre-filled syringe, for example, then you don't have an head space anymore. You can do that with your filling process, so you can also avoid that a little bit technically. So the polysorbates, however, this is the most used system, polysorbate 20, polysorbate 80 for that purpose. Also poloxamers to a certain degree have problems because they tend to oxidize, showing hydrolysis. And this is an example, and there are still, let's say, just one case of a problem that occurred, late stage phase 3 clinical trials already ongoing, close to the market, big pharmaceutical company right opposite of the Rhine River here, which failed because in the end, there was all of a sudden, after 1 year or 2 years of storage, particle formation. And the particle formation resulting either, and it has been shown, it can be even the free fatty acids resulting from polysorbate, but it can also be that the polysorbate degrades, and in the end, it does not do its job anymore. So this is the problem, and this is the stage where we said, let's use the phospholipids, look at the different phospholipids, different structures, and see how far we can get with those. Is there a potential to replace polysorbate in our formulations? So we use the different ones, ranging from the synthetic, purified 14 nil, 16 nil, and then going up and using more or less, and we started out specifically with the 18.2, which is sort of, the most easily and readily available material that we could get. You already heard a lot about hemolysis, so I don't have to introduce it much more, but this was right away, also from the funding side, a question, well, does it make sense at all if you want to inject the lysolipids? Aren't you asking for trouble? We said, yeah, okay, we think not, but okay, we have to demonstrate it. So what we did is we did hemolysis experiments, but now in the presence of serum, and in serum with the phospholipid bindings, with the lipid binding site, we see clearly that we have less hemolysis. Respectively, we need much higher concentrations of our surfactant to induce the hemolysis. Now, these concentrations are certainly lower than for the polysorbate or for poloxamer, where we hardly see any hemolysis. However, we have to rank that by the concentration that is required in our product. And this is what I'm going to demonstrate you, that we're going to use only concentrations, or we need only concentrations which are slightly above the critical micelle forming concentration, so maybe 0.1, 0.01 mg per mil, but we are much higher with the hemolysis concentration. So we're safe already, let's say, to look a little bit ahead, and that I don't forget to tell you in the end that hemolysis is not a problem. Don't get confused by this slide. It's purely illustration only, because we were looking at one hypothesis, which typically we refuse, is that our surfactant molecules would directly interact with our protein molecules. Long time ago, there had been studies and thoughts about pure red cell aplasia caused in erythropoietin products in the combination with surfactants, that these micelle structures could form, and this could be the reason. I mean, this interpretation was discarded in the end, but there was this interpretation that there could be also the interactions between protein and the surfactant. So we did CG mouse studies, and at Freiburg, supported by Heiko Herklotz and his team, we did ITC studies, and we do not see the binding of the phospholipids, as we would expect, to our protein drugs. However, we see it for the albumin. So does it work in the end if we want to protect our protein? 2 experiments. Left-hand side is shaking, right-hand side is freezing and thawing. Shaking on the first, a lot of blue graphs, but maybe it's only about one type of particles that I'm showing you, and you should have a look at the bars which are very low. So little particles is good. So good is if you don't use the MAP at all only, so that's bad. So good is the polysorbate, and also good are the higher concentrations of the phospholipids, except for the ones which is a pegylated version. It did not work as well. And you also see the concentration-dependent effect, that if it's above the critical micelle-forming concentration, then we see the effect much clearer. What we noticed in the freeze-thaw study, though, is that freezing and thawing, you may wonder why would you do it? There may be an accidental freezing and thawing, for example, of the product, but in many cases for proteins, we also have the drug product, which you have to store for a certain while, and this is typically also in the frozen state, and it's also the first part of a freeze-dried product, that you have to freeze the material. So what we did is freezing and thawing. We also see the effects of our lyso material, but we see still some high bars, and this is when we have too pure material, which is limited in its solubility, at a higher concentration even. So you may wonder with the 16.0 that it works at a lower concentration, but it does not work at the higher concentration anymore. It seems that we exceed the solubility limit. As we cool it down, it crystallizes, or let's say it solidifies out, and it is not capable of stabilizing anymore. So maybe a less pure material I may request for would be better. So then, okay, we knew it would work. It should have the similar effect. So let's confirm and see that it has also a film formation that is possible. So we see the decrease in the surface tension, and we did a lot of surface tension measurements. We looked at the elasticity of the film. I see that my time is already running out. So it forms it forms an elastic film at the respective concentrations. And also what we see is that depending on the concentration, if we move the barriers and we move the film and we are only a little bit above the critical micelle concentration, we have a challenge of filling up the gaps that we see in the film that is formed. We also notice that if we do that in the presence of the antibody, of our molecule of interest, that more or less we don't see much of an impact of the protein being there. Although what we see is that we have a significant difference, especially if we have a low concentration of our lyso material of the antibody on the surface tension. So we were not sure what is exactly going on, although it's overall a similar behavior for the lysolecithins, for the polysorbate, and also for the poloxamer, whereas the poloxamer forms the least elastic film. So we did ellipsometry. What in the ellipsometry we saw is that, yes, we have to start with a rather thin film. If we only have our LPC, the antibody forms a much thicker film. Those are the red triangles. But as soon as we have the mix in there already at a low concentration of LPC, the film thickness is almost right away at the thickness of our LPC film that is formed. That did not help us with respect to sensitivity, so we went up to Hamburg, to the DESY, to do some experiments there. And maybe specifically beyond the electron density that we can get from the jigsaw experiments, from the total reflection X-ray fluorescence, we can analyze the elements. So we can look at the sulfur, which is related to our protein. We can look at the phosphorus, which is related to our Lyso material. And that allows us now in the first, after the first measurements, to get some idea about the amount of protein that is still at the interface although the lipids already form their films. So we know that at the low concentration, for example, of the 14 nil, we are at maybe 8 molecules per 1,000 square nanometers, means that we still have about 1/4 or 1/5 of our surface covered by our protein. Higher concentration, we are below the limit of detection. Let me call it limit of detection. We still struggle with calibration and how we really can set it up and what it means, how far down we can measure. So the physical-chemical part, or the interface, we covered. Next thing was the chemical stability, and there was some interesting, we saw some interesting results, because the polysorbate, only look at the red numbers. The polysorbate, 25 degrees at different pHs, plus minus E means esterase. Esterase can be part of the esterase activity of the protein itself, or it could be something which comes from impurity, especially in early stages when you have some host cell proteins or something like that left in your product. And then you may experience, it's unstable, but it's actually the instability of the polysorbate caused by the less pure material. And this is what we see with the polysorbate. So without the esterase, it's fine, but then we also have some red numbers for our Lyso materials, and that we could show in the end with hydrogen peroxide experiments and the presence of methionine and plus minus esterase, because the esterase doesn't have an impact at all on the Lyso material, but the H2O2 and the methionine can rescue material. So the chemical instability of our 18.2 material is critical. And we also see some of for the pegylated version, which more or less was not that good anyways. So yeah, we ruled that one out. With the 18.2, we have to be very careful. And I mean, in the lifetime of a PhD student, you can only do let me call it still, long-term stability. So it's not 5 years, it's not even 3 years, because you have to start it at some point in your PhD thesis life. So it will run out for 1 year in total. We put different materials now on the stability, and we have a liquid stability now only short to see what the 18.2 can do for us. And we can clearly see that we do have, I say, in liquid, first of all, a kind of a stabilization. But what you see here is the particles, if you keep your product for 4 weeks at higher temperature and then shake. This is something that has been developed now in the course, also with regulatory authorities, that maybe in the end, after 2 years, you should show still that you have an effect of your excipient. And there you already see that it loses its effect. We don't see that, and the bar stays stable. That's the take-home message for both the liquid stability with the 14.0 study is ongoing. We will see what will happen after 6 months and 1 year. And the same thing also for a freeze-dried product, which is, let's say, the alternative that we want to use. So with that, I will summarize and say the hemolytic activity we consider as uncritical. The LPC stabilize the MAPs against our interfacial stress that can occur. The LPCs do not directly interact with the MAPs in solution. They form an elastic interfacial film. And with the sensitivity of the LPCs to oxidation and some of the solubility limits issues in the freezing and thawing experiments, we can come down to a few variants, probably the 14 nil, maybe in more or less purified 14 nil version, 18.1, 22.1, also options specifically also for freeze-dried product. Acknowledgements in the end. Thank you very much for the great support by the Phospholipid Research Center, Lipoid. I want to thank and specifically mention Eleni Papadopoulos, the PhD student doing the work, the whole team, Freiburg, DESI, TU Darmstadt, Wyatt supporting us also with some physical-chemical understanding and measurements. And thank you for your attention.
Thank you, Wolfgang, for this beautiful talk, which is now open for discussion. We will have time for one short question. One in the back, maybe I'm... Okay, Richard.
Yeah, thanks. Nice talk. I have a question regarding the selection of the lysolipid. So the intention was to substitute polysorbate with the lysolipids. Can you comment on the HLB values or like topological surface areas? Like how can the lysolipids be compared to the polysorbate?
We did not go by the HLB values because of the charge systems, so they are very different with respect to that. In the past, we had synthesized our own materials, so we used the sugar base, so you can think of sucrose, respectively trehalose, and attached a lipid chain to it, again, also rather pure materials with substantial hemolytic activity as well. They had been studied already also, I think in the 90s, maybe not in the 70s, by AstraZeneca as alternative surfactants. And with respect to the size we did and the occupied space at the interface, we did not consider that. We just wanted to see how. Let's say how we just had to learn by the CMC how much material we need with respect to that. So there's let me say a theoretical calculation. To some respect now with respect to solubility, it was trial and error, I have to say.
Yeah, but I think what would be interesting if you would look at the C-log-P and the TPSA, the topological polar surface area, and compare to the polysorbate. I would be curious like how these compare to each other.
Yeah.